Okay. And then Riley.
Here.
Okay. Riley Whistler.
Yep.
And then Riley Borden. I saw you raise your hand back there. John Brigo.
Here.
Okay. And JD.
Here.
Joshua. Yep. Kendra. Okay. Acacia.
Here.
Elijah.
Here.
Okay. And Daniela. Okay. Caitlin. Brittany. Kenyon.
Here.
Okay. Caleb Flores. And Graydon. Graydon? No? Okay. And Elia Gray. Okay. Sage Turner. Peyton. Rebecca Johnson. Okay. And Isabel Krause. John. No John today? Jenna Lunsford. Michael?
Here.
Yeah. Joelle. Here. And Lene Mink. Clark Moore.
Here.
Brooke Naylor. Chris Nichols. Benjamin Pringle. Ariana Villegas. Ariana? No. May. Okay. Kylie White. No Kylie. Okay. And Carly Ann. Yeah. All right. Anyone I didn't call? We good? Okay, today's the last day to enroll or drop without a W or anything like that, so just be aware. Okay. All right. Let's see, now that I teased you with the zombie, everyone wants one. Let's do.
I've never seen them before. I don't know why they're so cute.
Okay, so we are on to two phyla today. These are animal phyla. These are real animals, by the way. These are couldn't be confused. Oh gosh, I'm sorry for the non sequitur. Just popped in my head. I'm going to put this on as a link on the Canvas site. I was just yesterday reviewing news, and there is a really cool article about choanoflagellates that were found in Mono Lake. And this just came out as a press release yesterday. Pretty far out. And a creature the size of a dust grain found hiding in California's Mono Lake. Anyway, this is a colonial choanoflagellate that was found about 10 years ago in Mono Lake. And you can read the article. I'll put this link. It's just a one-page little news release. Anyway, so remember I was saying choanoflagellates have that collar-like cell in a flagellum that's used to feed. And it's a cell that's very, very similar to sponges. But I said the main difference between a choanoflagellate and a sponge is the sponge is multicellular, right? We have choanoflagellates, but we also have ctenocytes and leukocytes. Essentially, there is a division of labor, and that's what multicellularity is. That was kind of the main point. And I said choanoflagellates are usually colonial, meaning that they kind of live on stalks, but each individual cell is an organism, and they're responsible for feeding and reproducing themselves, right? So that's kind of like contrasting colonial life with multicellular life. So anyway, they found, so the story is, there was a student in the microbiology department at UC Berkeley who was an avid hiker and rock climber, and he was climbing in the Mammoth area and stopped by Mono Lake just to check it out. If you've ever been to Mono Lake, it's that lake, south, it's kind of south 95, south of Tahoe, and took like a water bottle, like a regular drinking water bottle, and just scooped up some water, and then took it back to the lab. They thought he'd just check out and see what kind of critters were living in Mono Lake, right? I mean, just random. And so they put he put the water bottle in the freezer, and it sat in the freezer for like a year before he ever got around to looking at it. So he pulls it out and finds these things, an undescribed species no one even knew Quantiflagellates lived on, right? This just happened, like I said, this just was published this year, but the sample was collected 10 years ago. And not only was it a new species to science, and Quantiflagellates were never even known to live in Mono Lake, they were all thought to be marine. I mentioned even last week that Quantiflagellates are all marine species. This is Mono Lake is a unique habitat. It's a very alkaline lake, kind of saline. So it's kind of a different type of habitat. But it's also an example of the first example of Quantiflagellates that have symbiotic bacteria. They have bacteria that live with them that help them do their thing, kind of like what a lichen is, or a coral. So it's a symbiosis between this Quantiflagellate and these bacteria to live in this weird kind of alkaline, salty habitat that's Mono Lake, only found there. So, just out of random chance, like scooping a sample and just looking at it, these folks are, it's probably going to be a huge scientific discovery. But anyway, the cool thing about it is this revived interest as the Quantiflagellates being like an example of what the earliest animals might have been like. So it just so happened it came out. I'll put a link. I'll put a link on Canvas about that. I can't think of a test question right now. I might ask about that, but the whole idea basically is that new scientific discoveries are being made every day. And sometimes you make a discovery without even knowing it, just collecting a scoop of water, sticking it in the freezer, and then just on a random lark, checking them out in the microscope, and poof, now this lab is probably going to be really well recognized and well funded to kind of study this because it's totally brand new, so pretty neat. Anyway, that aside, what I was talking about was that the sponges could probably just as easily be bumped into the kingdom Protista because they don't have tissues. The one thing that the Nigeria has that makes them a first important step to being a true animal is not only are they multicellular, they actually have true tissue layers and orientation, body design that has some orientation features. So we're looking at the phylum Nigeria, which includes the jellyfish, anemones, and corals, among others. And then I'm going to mention briefly the phylum Ctenophora. I don't have any good specimens of Ctenophora left. I think I might have some, but they're like so broken up. Yeah, here they are. They're so broken up and kind of messed up that I'm not going to have one test. But I'm going to show them to you anyway. So we have Kingdom Animalia. These are true animals because they have tissues and orientation. Kingdom Animalia. And then the phylum is Nigeria. Okay, so these are the jellyfishes, the anemones, the corals, etc. We'll get to the cone jellies at the end. All right, a couple things that are really important in this PowerPoint. This PowerPoint is available online, so snap a picture now, or you can just download this, to look at yourself at your leisure. But we're looking at some important things you have to know about this phylum. One is we have what's called diploblastic development. And what that means is we have true tissue layers. Diplo means two. Blastic means developmental, embryological development layers. And so we have endoderm plus ectoderm. So two tissue layers that we're going to talk about and why they're considered tissue layers. This is a really important feature of being an animal, is having tissues. They have symmetry. They have what's called radial symmetry. And this is where they have a mouth side and a non-mouth side. Or basically, they have sides to them. There's an orientation, a side where your mouth is and a side where your mouth isn't, right? Otherwise, they're kind of a round, wheel-like, pizza-like organism. They have a unique cell type called a cnidocytes. Cnidocytes are stinging cells, and that is unique to this phylum. Only this phylum has these kinds of cells, and it's a way they catch their prey. They're pretty much mostly predators. They catch their prey using these unique stinging cells that we'll talk about here. The rest of it, I think I'm going to go ahead and include in my diagram for the body plan. But I would say these three, the endoderm, ectoderm, diploblastic development, radial symmetry, and cnidocytes are kind of like the key things you got to know about the phylum overall. The remaining three, the mesoglea, the life cycle, the medusa versus polyp, and the gastrovascular cavity gut pocket, we'll talk about when we look at the design of the body. Okay? Sound good? Take a look. So, when we talk about symmetry, we're talking about the orientation of the organism. The sponge had no symmetry. There was no head, tail, right, left, front, back, just a ball or aggregation of cells. They were attached to a rock, but that was about the only orientation they had, right? So there's no orientation, no symmetry with the sponges. In a radially symmetrical organism, you have an organism that's kind of like a ring or a pizza pie. Well, they go ahead and draw out an organism here. We have two basic body forms that occur in the Cnidaria. There is what's called the medusa stage, and in the medusa stage we have a swimming organism. So let's see here. I'm trying to get some good coloration here. Let's try this out. All right. We have a medusa. It's kind of like an umbrella. It's like a here's kind of our medusa stage here, kind of like an umbrella or a hat or something. And the key feature of the medusa stage we're looking at is this is where the oral opening is. So this is the oral side. That's the mouth, basically. I mean, you could non-technically say mouth, but it's called the oral opening. And then the opposite side of the oral opening is called aboral. And all that means is non-mouth. There's no mouth on that side. So that those two sides basically set it up such that the organism, if you look at it from the top, is kind of like a ring. Oftentimes you'll see in these, I have a jellyfish, a moon jelly here in this dish. You'll see these kind of horseshoe-shaped structures here, and those are the gonads of the jellyfish. But then you'll see these rays radiating out to the outside skirt, called velum. But those rays are the feeding channels, the gastrovascular cavity that's distributing food to the outside. So the reason why this is called radial symmetry is I could take a knife or whatever, and I could cut it like this and have two symmetrical halves, or I could cut it like this and have two symmetrical halves, or like this, or like this, or like this. So basically, it's a round or radial symmetry. The only orientation is the mouth side and the non-mouth side. There's no right, left. There's no head, no tail. Just the side of the mouth and the side of the mouth, right? Makes sense. So this is what radial symmetry is all about, like a wheel. Okay, radial symmetry there. So we have a Medusa stage. This is called the Medusa. Of course, that's taken from mythology, because the Medusa was the, wasn't it like the lady that had snakes in her hair, you know, and you looked at her, you turned to stone. So the snakes in the hair thing was kind of like, because of the stinging cells, I think they have tentacles. That was kind of the idea. Anyway, the polyp existence is more of a sedentary, like a hydra or anemone style body plan. So here is the polyp. So in the polyp, and you've probably heard the term polyp in terms of like if you have a growth in your intestine, like in your colon, like colon cancer, usually starts as a polyp, which is like a little lump. Or maybe you've heard about singers that get polyps in their singing vocal cords, right? It's basically a lump or growth. The term polyp is basically a sedentary, like little bump. And you'll see some examples here. But the polyp stage is a sedentary style where there's an attachment disc and they attach to rocks or stick to a substrate using their aboral side. So this is the oral side, and this is the aboral side. And in this case, the aboral side sticks or attaches to some sort of substrate. Okay, that's the main difference. Usually these are associated with life cycle. Not always, but in general, the Medusa stage is the sexual stage, males and females, and the polyp stage is the asexual stage, where they reproduce asexually. That being said, all the first examples I gave you totally throw it out the window, and that's not true. So, but in general, the general theme here is that we have the sexual and asexual forms of the same organism. Okay, what else can I tell you? Oh, let's talk about this. So we have radial symmetry. You know what that means now. So you're gonna have to say that on the big lab exam somewhere. We have a polyp stage and a Medusa stage. You're gonna have to identify those and know what they are. Let's talk about this this development of tissue. So what happens is the body is made up of two basic layers of cell types that are forming the tissue layers. Why they're true tissues is because they have true epithelium. And what epithelium means is there's a basement, there's a membrane that separates that tissue from other such tissues. So it's not just an aggregation of cells, it's actually layers, specific layers of cells that have a basement membrane separated. So on the outside, we could say that this is called ectoderm. Okay. On the inside, the tissue layer is different. It's called well, we're going to call it an endoderm. I'll give you an alternative name for it that the book might mention, but I think we'll simplify life and just call it endoderm. Basically, it's a lining of the gut. We're going to call it endoderm. Sometimes it is called, in textbooks, gastroderm, which means stomach skin, or stomach tissue. We can go ahead and call it endoderm because that term is going to be used throughout the semester. But this group, gastroderm was the term used. I'm a big fan of not, splitting out all these terms. There's enough terms to know as it is. Why do we have to learn unnecessary terms? So that's why I have the also known as gastroderm for the endoderm. On our test, you can say either one, but endoderm would be easier for you to remember because we're going to use that again down the road. Okay. Let's take a look here at how this works. I'll take like a view here. I'm going to zoom in like I'm zooming in on a microscope. And the endodermal cells are kind of columnar. So here's the endodermal cells. And these endodermal cells are ciliated, meaning they have cilia, kind of lining the inside layer. And what those cilia do is they generate a water current or circulate fluid in that cavity. So these are cilia. Ciliated, and then this is the endoderm. Okay, and then the outer layer is ectoderm, and then occasionally there will be a cnidocyte on here. These are kind of squamous cells, like flattened cells. Occasionally there'll be like a kind of a spherical cell. So this is ectoderm. And these cnidocytes are the stinging cells that occur here. These are cnidocytes. Cnidocytes meaning literally stinging cell. Sometimes you will hear the term nematocyst. So cnidocytes is the more accurate way to describe the cell. The nematocyst is the structure in the cell that does the stinging. So I don't really care. I'm not going to ask you to differentiate those two on a test. I should, but I think they're related terms. So cnidocyte is the correct answer, but if you wrote nematocyst, it would be wrong. It's just that nematocyst isn't describing the cell, it's describing the stinging structure. So here is the stinging cnidocytes that occur on the outside of a hydra tentacle. And you can see the little, like a little harpoon looking thing on the inside. Here's one right there, you can see the sharp harpoon. What happens is there's usually a trigger on the outside of the cnidocyte. And when that's triggered, the cell becomes very hypertonic, water rushes in, and the cell explodes. And launching out of the cell will be this harpoon-like thread that pierces the prey that's trying to catch. And then usually there is a very, very potent venom that's associated with that cnidocyte and nematocyst. And that venom is usually a neurotoxin that will prevent the prey from thrashing around too much. Because these things are only 2 cell layers thick. If like a jellyfish is catching a fish and the fish thrashes, it could break the jellyfish's arms or tear a jellyfish up. So the neurotoxin is to subdue their prey very, very quickly. Get the idea? Okay, so the cnidocytes occur on the ectoderm. That's the main thing. Then there is a jelly layer that occurs between the two. And this jelly layer is called the mesoglia. And this is the jelly of the jellyfish. This mesoglia layer. Okay, so the mesoglia layer occurs between the ectoderm and endoderm. There's no cellular component of that, although there are amoebocyte-like cells that would run around there. So let's go ahead and draw a similar example here for the Medusa stage. So here we have the endoderm, and then usually the Medusa stage, the jelly is the mesoglia is more prominent. because the mesoglea helps in buoyancy, it helps it float. Okay, so this is the mesoglea. Here. Okay. There is a dish that's popular in Southeast Asia, like in Vietnam and Thailand, and it's called a jellyfish salad. So they'll take moon jellies like this one, fresh, and they'll pan fry it in vinegar. And pan frying it in vinegar pops all the stinging cells, the nematocysts, so they don't sting you. And anyway, then what they'll do is they'll slice it up into thin slices like you would like a mushroom, and they'll put it into like a noodle salad. And it's really interesting that the mesoglea, the jelly, actually is crunchy. It's kind of like, it's not like Jello. It's like eating celery. It's weird. So it looks gelatinous like Jello, but when you eat it, it actually kind of has the same kind of texture as celery fruit fibers. So anyway, if you ever get a chance to eat jellyfish salad, I don't know if you can get it ready, but I'll be in San Francisco. Try it out. Okay, we're looking good. Oh, one last feature is the gut cavity. The gut is called the gastrovascular cavity. This is important. This is called a gastrovascular cavity. So this, I'm not going to let you just call it a gut, because there's all kinds of guts. This specific style of gut is a gut pocket. And it's called gastrovascular because it's a stomach, and it's also a circulatory system. It's a circulatory cavity as well. So what happens is the food is brought in, it's digested, like they'll eat a fish, they'll swallow the fish in the mouth, and then enzymes will be secreted onto the fish that will break up the food into smaller particles, and then endocytosis or feeding can happen in the cells. So these are able to eat prey that are much larger than individual cells, which is totally different than a sponge. So that's the gastrovascular cavity, and then this one also has a gastrovascular cavity. Gastrovascular. Or we could probably the better incorrect way to describe this is a gut pocket. So we'll call this a gut pocket. I want you to know the term gastrovascular cavity because several phyla have this, whereas everything else this semester will have a gastrointestinal tract. So you have a gut tract, a mouth, a tube, and a hand, and so on. Okay, what else can I share with you here? Let's take a look at classification. So we have 4 different classes of Cnidaria to think of. I only have examples of 3 of these. I don't have the cubozoa. But I'm going to mention the cubozoa because you've probably heard about it, so you can think about it if you ever go to areas that I might be. Phylum Cnidaria, and the class Hydrozoa. Okay. The class Scyphozoa. Okay. Class Cubozoa. And again, I don't have this one. And then the class yeah, you will have to know these names: Hydrozoa, Scyphozoa, and Anthozoa. And usually the question comes up: do I have to spell it correctly? Of course you do, right? And you've got to spell it right. So it might be worthwhile for you to have somebody make flashcards or whatever, or to practice writing these things out so you don't butcher the spelling. Okay. Is it wrong if you spell it wrong? Depends. Okay, so I'm not going to commit to anything. But if I can understand what you mean, I'm going to give you credit for knowing the organism, but maybe half off or whatever, half point off for misspelling. So it's in your best interest to film things, right? All right, so here we have these 4 classes. There are some other classes that are kind of rare and might be obscure, so I'm not even mentioning them, but these are our main ones here. Okay, so what I'm going to do here is I'm going to go through each class, point out the specimens that we have, let you know what I want you to know about these things, and give you some hints or suggestions for the test questions, right? That's kind of the plan here. So let's start off with the hydrozoa. Probably the one that people most know here is the hydra. And that's a genus. We also have Obelia. And we also have it's Physalia. Okay, again, I'm not going to demand that you know these genus names, but you know, if you see this sitting on a test, and there's a little label there that says Physalia, that might help you, right? I mean, you know, it could be a little hint as to what the thing is. But these are the 3 examples of hydrozoans we have. The hydra is here, the Obelia is here, Obelia's here, hydra's here, the Obelia's here, and then the Physalia is here. Okay? All right, let's take a look at hydra. I think the hydra, pretty much, this is an example of what the hydra looks like. These are freshwater and marine species. They only exist as a polyp, even though I said that there's an alternation of generation oftentimes. The hydra only exists as polyps. So you see them as a stuck stage like this. I have 2 slides that you should look at. You should look at the whole mount. WM equals whole mount, which will look like this. And then I have a CS, cross section, which will look like this. Okay, so in the cross section, CS, they've taken the whole mount and they've cut it across the body or across the arms. So what you're seeing basically in the cross section is you're seeing the endoderm and ectoderm, and then you're seeing cnidocytes. So be sure to look at hydra nematocysts or hydra cnidocytes. Those are cross sections of the body or the arms. What you're going to see in that is something like, let's see here. You're going to see basically, ultimately what looks like this. Here's the arm, and you're going to see a cross section through it, and you'll see the ectoderm and the endoderm, the gastrovascular cavity, the mesoglea. It's likely that I will have a slide that has a pointer pointing right at cnidocytes. I'll ask you, what type of cell is this? Cnidocyte's the best answer. You know, a number of things there. So I don't have a good image of what you're going to see. But basically, the cross section is going to be the close-up view of the body wall tissues. Okay. Sound good? All right. So that's the hydra. So look at 2 things. We have a whole mount, it's like this, and then we have cross sections and long sections will basically be, you can see the tissue layers. There's 2 piles of slides there. Obelia, this one you should look at the polyp colony. And the medusa stage. I am looking at my pile here, and I'm seeing very, very few of the colony ones. So I'm going to dig through these boxes to see if I can find some more. I only see like 3 or 4. I see 4 slides of the polyp colony. That's kind of disappointing. I think what happens is over time they get broken or they get old. or the cover slip crack because of the light and the air. They have a shelf life, you know. But anyway, you're gonna find one on the test, right? So that being said, we have almost 30 people here. Not everyone's gonna have one to sit at their desk, you know, all 2 hours. So share on that one. Okay, the polyp colony is a limited supply of those. There's more medusa available. But this is what they look like. So in the polyp colony, we have basically a polyp, but it's a colonial polyp, meaning it's several individuals live on the same stalk, basically. And the feeding polyps are here with tentacles. The tentacles have little cnidocytes, they have stinging cells they use to feed on microscopic organisms, little protists and such. They have their own little gut pocket, etc. So what happens is when they're immature, the polyps are a feeding polyp. But then what happens is once they mature, they become a reproductive polyp called a strobile. And the strobile is basically the reproductive stage of this organism. Let me go ahead and draw this real quick. So just so I can tell you what the questions are going to be on the test, right? So on the new ones or the young ones, we have these feeding polyps. The ones with tentacles are immature, and these are basically feeding polyps. Okay, and the ones that are more at the base look kind of like this. And the technical term is a strobile. And you've probably heard this term in botany, that like alder seed pods or seed cones, flowering plant seed cones are called strobile in botany. So basically it's kind of like referring to this as like a cone, right? And a cone is a reproductive structure in a plant. The strobile is basically an asexual reproduction pod, and each one of these little rings is a baby medusa. So what happens here is out of this cone, what you'll have occur is a medusa stage. Okay. So the strobile produce asexually medusa. And then the medusa go out into the water column to swim and disperse. And then they are sexual reproducing organisms. The medusa basically will sexually reproduce, and they alternate between medusa and polyp stages. Right? So it's kind of like a little alternation of generation, although they're the same ploidy number, not like plants. They maintain the same chromosome number, but they basically alternate between polyp, medusa, and so on and so on. Okay? Sound good? All right. So on that obelia slide, I want you to see an obelia colony. I'll probably have a pointer pointing to a feeding polyp and something else pointing to a strobile, and I'll ask you what the purpose or function. You should know they're polyps, and you should know that the feeding polyps have tentacles, and then the strobile, which have the little rings in them, each of those rings are basically a baby medusa. And when they pop out, as they are here, that will basically mature into the sexual stage, which is the medusa stage. I have medusa here. The ones I have on the slide look kind of like this. This is where the mouth is, called a manubrium. And then the umbrella part is kind of popped up. You know, if you have an umbrella and the wind catches it, how they can be inverted? That's kind of what that image is showing. This normally would be like this, but what's happened is the little edges of the dome are flipped up because in the preservative they use, it kind of destroys the body. This is a good example of radial symmetry here. And these structures are gonads. So there's one thing about what makes the hydrozoa a unique group, even though that looks like a jellyfish, it's not a true jellyfish. The definition of hydrozoa is they have ectodermal gonads. The gonads are on the outside of the body. So you'll see on some of these hydra, they'll have spermaries or ovaries produced on the outside. And that's what's going on in this producer stage, is these gonads are actually on the outside, adjacent to the oral opening, the outside opening. But kind of cool. That's what defines the hydrozoa, is the ectodermal gonads. Everything else has endodermal gonads, they brought it inside. Okay, so you know what you can see there. Lastly, we have Physalia, and this includes a couple species. There's also a related species that you might see in California. These are called colonial polyps. They're floating colonial polyps. And so in the Physalia, what's going on is, let's see here. It is the one individual polyp produces the gas bag. So this is this is a polyp here, so it's basically like a little hydra here. But attached to the aboral side, there's the oral side, is this gas bladder, and that's what makes the floating part, or the sail. So this is called the float. This is the float here. So that individual makes the float, and then they asexually produce more feeding polyps on there. So there'll be basically like, a dozen or more feeding polyps. And some of these feeding polyps can have tentacles as long as like 20 or 30 feet long. So what they do is they float on the surface, the tentacles hang down like 20, 30 feet into the water, and they catch small fish using very, very potent netocytes and draw them up. But basically, it's a colonial polyp. And there might be some sexually reproductive polyps as well. But the main thing with the Physalia to know for the test, and I have that one in the jar there, is this is a colonial polyp, where one individual makes the float, and then the rest of the individuals are kind of hanging on the float together like a little apartment building. But it's not a jellyfish at all. It's a polyp stage that's floating. Okay. The Physalia occur in the Pacific and the Atlantic. Surfers have a problem with these because what happens is they get blown to the shore, and then when you're surfing, the Portuguese man, or what it's called, or sea wasp, will get rolled in the waves and broken up, and so you'll have like a tentacle that's like 2-3 feet long by itself, and so you're surfing and the tentacle hits your arm, and you get a big sting wound, basically it's like someone whipped you with a piece of barbed wire or something. Usually the Physalia won't kill you, but what happens is the people who are overwhelmed with that, like they get the tentacles across their body, can panic and drown. It's usually how you die. But that's the way it goes. Questions? Good? We also have one here like in Sonoma County and Marin County and Mendocino County. These will blow up on the shore usually in the fall. They're called by the wind sailors. They're like a little mini version of a Portuguese man of war, but they're not really poisonous. Although if dogs are on the beach and they go around and lick them, the dog can get stung or they get their nose stung. So
I was gonna say, don't they?
the way it goes.
Too.
Okay.
Yeah.
Any questions on these? So Physalia, I have that specimen there. Okay. I think I have one more in the cabinet as well. Scyphozoa. These are the true jellyfish. And the defining feature is they have endodermal gonads. The only one I have here to look at is Aurelia, which is the moon jelly. But I have actually multiple life stages of Aurelia. So in Aurelia, I have the life stages listed out here. Again, the Medusa stage is the sexual stage. The Medusa is in this dish. It's the jellyfish basically. It's quite sizable, right? These things can get as big as this dish itself. These are very, very small ones. But that is the sexual stage. So there is the Medusa. The rings, the little horseshoe structures, are the gonads. And they're found inside the gastrovascular cavity pocket. So one question I like to ask, so I'll have that jellyfish dish, and I'll say, what are the horseshoe-shaped structures? And those are the gonads. Okay. You can't really tell if they're eggs or if they're ovaries or testes. Kind of look the same. But basically, those are the tissue structures, and they're found inside the gut pocket, which is what defines them as skytosaur. Okay. Okay. So, but there's also some other life stages. What happens when they reproduce? The sperm and eggs get together, and the developing zygote becomes. And I'm just going to let you find them on the slides here. So I'm not going to draw this out. They're all there on the slides. There's a planula, which is like a little larvae stage, it looks almost like a paramecium. And then these stages here, all three of these, are called scyphostomum. The term stomum means mouth. So these are the feeding polyps. These are just the feeding polyp stage. They have tentacles. Then the feeding polyps become what's called a strobilus or strobile, like this. And this is basically like a Pez dispenser for Medusa. Pez dispenser, right? You pull the head back, and a little Pez comes out. So it's the kind of thing, you pull the little head back and a little Medusa pops out, right? So this is like a stack of Medusa here developing like a cone, and they'll become basically baby Medusa. The baby Medusa is called an Ephira. I'm not going to ask you these names, but on the test, it might say Ephira on the label, and that one you can ask you what that is, right? And the Ephira matures back into the Medusa stage. So it's Medusa, planula, feeding polyps, strobile, back to Medusa, right? It's kind of the idea. So it's the same basic idea as this, but it's not a colonial thing, it's very individual polyps. So those are all here. It's a little overwhelming because there's all kinds of names. What I'd like you to do is to kind of associate who's who. And guaranteed I'm going to have on the test the Medusa stage, the adult Medusa, right? In the dish, just like this. But I might, ask you about the feeding polyps versus the strobile, etc. So just take a look at those, okay? So just kind of think about that. Don't get too bogged down in memorizing all of them. But the main point is to get the idea that there's Medusa stages and polyp stages. The polyp stage is the asexual reproduction. Medusa stage is usually the sexual reproduction. Some Scyphozoa can get pretty big. There's a big lion's mane jellyfish, huge. They're not super toxic. Some of them can be a little bit more poisonous, but these are just kind of moderately dangerous types of jellyfish. On to the real dangerous ones, though. The Cubozoa. Okay, let me just go ahead and write this here. So for the Aurelia, you should know the Medusa, feeding polyps, strobile, etc. So basically, you should know the life cycle of the Aurelia. What I want you to know on that. For sure, the adult Medusa will be on the test. Okay. Cubozoa, I don't have any specimens, but you should know about them. They're called the fox jellies. And the reason why they're called fox jellies is because their body, their gastrovascular cavity is kind of four-pointed, or four-parted. So these are usually the size of a golf ball in nature. And the gastrovascular cavity is usually kind of like four-sided. And so there's usually just four tentacles that stick down from those 4 points. And so that's where the name box jellyfish comes from. What you should know about it, they're the size of a golf ball or a ping pong ball, but the biggest species can be the size of a baseball. The biggest thing you should know is this is one of the most venomous animals on the planet, and causes quite a number of deaths, like in Australia and South Pacific. So they're predators of small fish, and the neurotoxin is super potent, such that when it hits a small fish, it immediately paralyzes it. So what happens is people will go swimming, and of course the box jelly is not trying to eat you, but you brush into it or bump into it. And again, surfers, what happens is they get into the waves, and the box jellies are getting kind of tossed around in the waves, maybe even broken up, and you get these horrible sting scars on your legs. Usually what happens is if you get it on your chest, it can paralyze your breathing. Basically, you stop breathing and you drown. That's how they can die. There's a movie with Will Smith called look at the movie, box jelly is a major star. Yeah. 6 pounds. Yeah, that's right. And the box jelly basically makes a major role in. Should I spoil the punchline? You want to watch it? Anyone want to tell us why the box jellyfish was a major character? What's that? Do you guys know? I'll let you watch. Now you want to watch. Okay. So anyway, Will Smith's character, he keeps a box jellyfish as a pet for a specific reason, which is really interesting. Okay. All right. So, class Anthozoa. These, here's some features. They exist as a polyp only. There is no medusa stage in the Anthozoa. They only exist as a polyp. They also have a what's called a folded internal gastrovascular cavity. Their gastrovascular cavity is folded, and this lets them have more bulk or mass. So most of these things are only 2 cell layers thick. So in order to get bulk, folding that 2 cell layers thick is kind of the only way. So here is one that's cut open, and I have one over here cut open. So it's the same basic idea. Here's the oral opening, here's the tentacles. Here is the gastrovascular cavity, and then here's all these folds. It looks like a stomach bone, like tripe. And you have these kind of folded internal gastrovascular cavity pocket, which helps make the organism more bulky. And then these granular material down here is gonads. All the gonads are basically found in the stomach. And so we have a sea anemone is an example of these. And so here's our sea anemone. And you should note the folded gastrovascular cavity, the one that's cut open, and then you should note that they have radial symmetry and that these exist as a polyp. The 2 common ones that we see on our coast here is the what's called the elegant sea anemone or the colonial sea anemone. These are all over the rocks, like in Trinidad, and if you touch them with your hand, they kind of squirt, the water squirts out of their gastrovascular cavity. Underwater, when the tide's up, they come out and feed, and then when the tide's down, they kind of hunker down when drying out. There's also the giant green anemones and the tide pools, but these are basically common sea anemones as an example. So the one I have here is, its genus name is Metridium. Again, it's no big deal, but genus names. So these are Metridium. And then I have some slides here of Metridium long sections and cross sections. And in those long sections and cross sections, you can see that folded gastrovascular cavity and the gonads inside of the gut pocket. Got it? So here's the slide, and then here's the actual critters. And then we have coral. And remember, coral is a symbiosis of a basically an anemone plus a algae plus a zooxanthellae. So we have basically the zooxanthellae are the symbiotic algae dinoflagellates that live in the mesoglia of the coral polyp. So here's the polyp here, and then here are the algae living with it. And what the algae does is the algae grab carbon dioxide and calcium from the water and make that really hard cement-like limestone coral. So what the algae does is the algae makes this hard structure, and then the polyp lives in little pockets. So if you look at this coral, you'll see these little, what almost look like little holes in the coral, and that's where the polyp was. The polyps are very, very small. Here's a coral in preservative, and you can still see the polyps in the fluid hanging out with little tentacles, but they're not very big. They're like a couple millimeters in size. Got it? So be sure that you remember we already saw the coral. So on the exam, I'm going to ask you, a coral is a symbiosis. It's basically a symbiosis between an anthozoa and a zooxanthellae. Is kind of the main word. An anthozoa and the dinoflagellata is basically a better example of what it is. But there's the answer. Coral equals anthozoa polyp plus a dinoflagellata algae. That would be the best answer to the test question. Got it? Okay. Anything else? Look good? Question an hour here. Coral bleaching, you might hear about this like in environmental science news. What happens is the algae needs sunlight and carbon dioxide to thrive. And the polyp benefits from the house that's made by the algae, but the polyp feeds itself using tentacles and stinging cells. What happens is when marine pollution happens, usually what happens is the water gets warm or the oxygen levels get lowered, is coral will lose their algae, and it's called coral bleaching. And it's kind of a crisis in tropical zones. It's the equivalent of what you think of with like deforestation in the rainforest. Coral bleaching is a major crisis because it's kind of the root of the food chain. Coral reefs are the basis of the food chain in tropical oceans. And so it's a major problem. So it's called bleaching because basically they lose their photopigments and then the polyp dies. And what's interesting is the algae can live without the polyp, but the polyp can't live without the algae. So it's kind of like a classical codependent relationship there. So there you go. There's a cool article in Nature about bleaching if you want to check it out. And finally, again, I don't have a specimen of these, but I think it's worth mentioning. So I won't have these on the test, because I don't have a good specimen. But the phylum Ctenophora is a related phylum. They're called the comb jellies. And the reason why they're called the comb jellies is these are usually kind of football shaped. They're the size of a golf ball or a baseball, and they're like kind of the shape of a football. They kind of look like this. And what's interesting about them is they actually have a rudimentary complete gut tract. So their gastrovascular cavity actually has two openings, which you could call an oral side and an anus side. It's kind of a mystery as to how this is because it's totally different from our gut. But the main thing I want you to kind of know about these, or don't really have to know it because I don't have any specimens. But they have these rows of what are called combs. And the combs are called colloblasts. Colloblasts. And what colloblasts are is they're sticky cells. Rather than stinging their prey, they basically shoot out flypaper, glue, and they will attach to their prey using a sticky glue material, and then draw it back in and feed. So that's where these names, the combs, come from. These are usually in rows along the body. But they're a group that is superficially related to the Cnidaria because they share primitive traits. Ectoderm and endoderm, gastrovascular cavity. But again, remember we can't classify organisms based on sharing primitive traits. We've already showed that's not a good idea. So we go ahead and put them in the same day because they share a lot of the same primitive features. But again, I don't have any specimens, so we don't need to worry about it. There you go. All right, there we go. An hour's gone by. Any questions? I think everything's out here. You probably want to get a picture of these anemones in the dish, and you want to think about the questions that will be asked going backwards. I have, some coral here, and I'll certainly ask you about the symbiosis of what this is a symbiosis between. So I'll probably have like some hard coral on the test and ask you what organisms contribute to making that, which would be the Cnidaria class Anthozoa plus a dinoflagellate algae live together to make coral. I have the Anthozoa anemone here, and it has the folded gastrovascular cavity. It's also gonads in the gastrovascular cavity. Probably I'll have the big one. I probably won't have the slide on the test, but I'll have the one in the dish. Okay. I have the Medusa stage of the Aurelia. That'll for sure be on the test. I'm not sure I'm going to have any of the larval stages of the Aurelia, because I already have the Obelia that has the feeding polyps, Cerobilia, and Medusa, right? So I think that probably could be good. I have this manowar in a jar. That's one of the only ones I got, so if I'm going to test, think about what questions will be asked. It's in the class Hydrozoa. It's actually a colonial polyp, meaning multiple individuals attached together, but they each have to feed themselves, and they're all attached to one gas bag. And then I have Hydra, and full mount specimen show the polyp stage, and most likely you're going to see though is the cross section that'll show the endoderm, ectoderm, and metacytes. All right. Cool. All right, that'll be it for today for my blabbing. So you got an hour plus to check out these specimens. I do have all the slides from last week, but we're kind of like beyond that, right? So you don't want to get bogged down. What do you need from last week? Let me know and I'll get it for you. I'm not going to put them out this time though, because that's dangerous.